Students, we are going to discuss about an important cellular organelle, ribosome. Ribosomes is a tiny granular particles of ribonucleoproteins which serve as the factories of protein production. They are present in all prokaryotic and eukaryotic cells. Ribosomes were first discovered by Romanian cell biologist George Emil Palade using an electron microscope. This organelle was first named as microsomal particles, but in 1958 Roberts introduced the term ribosome. These are the cell's factory, that is the site for protein synthesis. The process for protein synthesis is known as translation, so that it is also known as translational apparatus. These are non-membranous organelles, mostly attached to rough endoplasmic reticulum and also found free in the cytoplasm. It is also made up of ribosomal RNA and ribosomal proteins. These are of two major groups, prokaryotic and eukaryotic ribosomes. Eukaryotic ribosomes are of three classes: cytoribosomes, mitoribosomes and chlororibosomes. Cytoribosomes are present on the cytoplasmic matrix. Mitoribosomes are present on the mitochondrial matrix. Chlororibosomes are present on the chloroplast matrix. Eukaryotic ribosomes are of two groups: freely dispersed ribosomes and attached ribosomes. Freely dispersed ribosomes are randomly distributed in the cytoplasm, whereas attached ones remains attached to rough endoplasmic reticulum, mitochondrial membrane and nuclear envelope. Membrane-bound ribosomes mainly involved in the synthesis of proteins. For membranes, it also helps in exocytosis. Free ribosomes synthesize proteins that function inside the cytosol. That is, it also helps in food metabolism. The average sedimentation value of these ribosomes are measured in Swedberg units. Next, structure. All classes of ribosomes have almost the same structure. Typically, they are porous and spherical particles. Each of them consists of two subunits, one larger and other smaller. The subunits of the ribosome are synthesized by the nucleolus. The larger subunit is almost dome-shaped and the smaller one is placed one above the other. It is just like a cup arrangement. In between these two subunits, there is a groove present. In this groove, the mRNA is positioned during protein synthesis. In this groove, the mRNA is well protected from the degradative actions of ribonuclease. Ribonuclease is a type of nuclease enzyme that catalyzes the degradation of RNA into smaller components. The 70S prokaryotic ribosome has 50S and 30S subunits. The 80S cytoribosome and the 78S chlororibosome have 60S and 40S subunits. The 55S mitoribosome has 40S and 30S subunits. A ribosome is mainly engaged in protein synthesis. It have 4 binding sites for the binding of mRNA and tRNA. the M site for the binding of mRNA, and P site called peptidyl site that binds to the tRNA that holding the growing polypeptide chain of amino acid. Next, third site is A site, that is the acceptor site. It binds to the amino acyl tRNA which holds the new amino acid to be added to the polypeptide chain. And E site is the site, that is the exit site, through which the uncharged or unloaded tRNA is binding. അതായത് 4 സൈറ്റുകളാണ് റൈബോസോമിലുള്ളത്. എ സൈറ്റ്, പി സൈറ്റ്, ഇ സൈറ്റ്, mRNA ബൈൻഡിങ് സൈറ്റ്. പ്രോട്ടീൻ ഉണ്ടാവുന്ന സമയത്ത്, അതായത് പ്രൊഡക്ഷൻ്റെ സമയത്ത് mRNA ഈ 2 സബ് യൂണിറ്റിൻ്റെ ഉള്ളിലുള്ള ഗ്രൂവിൽ അറ്റാച്ച് ചെയ്യും. mRNAയിൽ നിറയെ കോഡോൺസ് ആണുള്ളത്. ആ ഓരോ കോഡോണും അമിനോ ആസിഡിനെ റെപ്രസെൻ്റ് ചെയ്യുന്നു. അങ്ങനെ ഇൻകമ്മിങ് അമിനോ ആസിഡ് എ സൈറ്റിൽ വന്ന് ബൈൻഡ് ചെയ്യും. പി സൈറ്റ് എന്ന് പറഞ്ഞാൽ പോളിമർ സൈറ്റ്. അവിടെ വെച്ചിട്ടാണ് വരുന്ന അമിനോ ആസിഡും ബാക്കിയുള്ള അമിനോ ആസിഡും കൂടി ചേർന്ന് പെപ്റ്റൈഡ് ബോണ്ട് ഫോം ചെയ്തിട്ടുള്ള പോളിമർ ഫോം ചെയ്യുന്നത്. ഈ സൈറ്റ് എന്ന് പറഞ്ഞിട്ടുണ്ടെങ്കിൽ അവിടെ വെച്ചിട്ടാണ് ഇപ്പോൾ mRNA കോഡ് ചെയ്തിരിക്കുന്ന എല്ലാ അമിനോ ആസിഡും വന്ന് ബൈൻഡ് ചെയ്ത് ഉള്ള പോളിമർ ഉണ്ടായിക്കഴിഞ്ഞാൽ ആ പോളിമറായിട്ടുള്ള പ്രോട്ടീൻ എക്സിറ്റ് ചെയ്യുന്ന സൈറ്റാണ് ഈ സൈറ്റ്. The 2 ribosomal subunits remain bound to each other during protein synthesis. Soon after protein synthesis, these 2 subunits dissociate. Their association and dissociation depends upon the magnesium ion concentration. There are 2 critical levels of magnesium ion concentration, upper critical level and lower critical level. In upper critical level, promotes the close association between the ribosomal subunits. If the magnesium ion concentration falls below the upper critical level, the 2 ribosomal subunits dissociate. And if it falls below the lower critical level, each subunit disintegrates into its constituent molecules. If the magnesium ion concentration goes well above the upper critical level, the 2 whole ribosome may bind together and form a dimer.